So Margareta Lukusic, I'm not sure if I pronounced your last name right.
Lakusic, but it's okay. Yes.
Lakusic.
Yes.
She's a PhD student in University of Porto in Portugal, which I think is where Ignacio did his degree as well.
Exactly.
But Margarita did her undergraduate and master's degree at the University of Belgrade in Serbia. And I know that she's quite interested in snakes because I met her a couple of weeks ago in America, in Portland, at the GRC meeting. And she had a lovely poster about a very cute little viper species. And I suspect that you will be talking about this species today. So without further ado, thanks for being willing to give a talk. And the digital podium is yours.
Thank you so much, Ronald, for the lovely introduction. You took a bit of my first slide, but this is perfect. And thank you, everyone, for being here and for attending this almost last, unfortunately, talk under the scope of the cost action. And as Ronald said, I am Margareta Lakusic, coming from Serbia, doing a PhD in Porto. My PhD is supervised by Fernando Martinez Ferreira from CBio, Juan Calvete from the Institute of Biomedicine of Valencia, and Benjamin Florian Hempel from the Freie University of Berlin. And for my PhD, I'm interested in studying. I'm sorry, I just need to figure out. Okay, well, I have part of my screen covered. It will move out to the banner.
We can see your entire screen.
Okay, but I'm not seeing it because of the Zoom banner that goes over it. So, do you know? Okay, it moved. Let's keep it like this. I know what I will talk about. So I'm interested in studying the evolutionary and ecological factors that affect snake venom variation. And when we think about venom in terms of the snakes, it's a bit of a link between the animal itself and its environment. Therefore, it's frequently observed that there is a coevolution between the expression of specific toxins and different diet composition. And this phenotype, this highly complex phenotypes, is highly variable. We can see many variations between different species, but also at the level of one species as well. So when we think about between individuals, between juveniles and adults, between different sexes, males and females, between, if we compare the venom from originating from different phylogenetic clades, and also when we study the venoms that are found from the populations under distinct ecological conditions. However, these factors are not, at least until 2021, when Mike Damm and colleagues published this work, these factors were not considered frequently in this in the aspect of studying snake venoms. I would say that now in the last years, there is a huge improvement in that, and this orange part is being decreased. And this is where I would like to contribute a bit and to increase the knowledge about the venom of nose-horn viper. So this is a Vipera mellitus species complex. As you can see on this map, it is presented with many different phylogenetic clades from North Italy across Balkan Peninsula to Asian Minor. In terms of feeding ecology, there is difference as well. There is a difference in diet preferences between juveniles and adults. So there is an ontogenetic shift. The diet composition with juveniles feeding predominantly of ectotherms, lizards, and sometimes centipedes. And as they grow up, they include more of the small mammals in their diet. When we compare just the males and females, well, the previous studies that compared males and females, they've shown that there are no differences in the prey preferences between these two groups. Once there is the snake bite by this species, this snake bite can cause hemorrhage, necrosis, and neurotoxicity. And based on the wide distribution, based on the clinical picture, sometimes can be highly complex, and even sometimes there are lethal outcomes of the bite of this species. World Health Organization classified it as a species of the highest medical importance. Also, this attracted a lot of interest in studying the venom, the anti-venom, the clinical pictures of it. And one of the studies showed that even the currently two available anti-venoms are having different level of efficiency against the venoms from different populations, from Macedonia, Montenegro, Bulgaria. They are differently in a different level effective. So all of these draw a lot of attention, also to understand the composition of the venom itself. Therefore, there are already some publications on different phylogenetic venoms from different phylogenetic clades, and most of them showed out that the major toxin families are snake venom metalloproteases, PLA2s, SVSPs, and C-type lectins, which is a composition that is dominated in many other viperine venoms. However, despite seeing that this venom is highly studied, there are still some questions that could be replied and well posed and hopefully answered additionally. For example, there are no studies on the local populations closely geographically located, but under distinct ecological conditions. Or also the comprehensive study across the species distribution is missing. So therefore, I found some of the questions that could be hopefully addressed throughout my PhD. And this is actually, so this is more or less the part of how and why I started studying the Vipera moditis, but how UVen helped in all of this and what was the role of the cost action. So it was 2020, I moved to Portugal. Actually, I lived with Ignacio as a roommate. We were in the COVID time stuck a lot in front of the computer. I opened Twitter account, and in those days there was an announcement of UVen action that is now being just set off to start with a lot of invitations to include the early career researchers with the announcement of the seminars. And since it perfectly aligned with what I was working on and what I was interested in, I applied to 2 working groups. It was the Novel Targets in Venom Research, Working Group 1, and Working Group 5 that focuses on training. So in the beginning, I was mostly a passive participant in the cost action. I attended many online seminars, the first international congress, and also the introduction in economic training school. And all of this for me, as again the first year PhD student, was amazing because I got the full idea, not full, but highly broad idea of what is going on in terms of the research in Europe and broadly, who are the people working in the field. And throughout attending these courses in school, I learned also a lot about the techniques, how to analyze the data, or at least I got the first idea on how about the data analysis, and also what are what's possibilities in the application of venom studies in pharmacology. So this is still a bit of online time. In that time, I met Mike Damm, who probably many of you know, and he was the one, and he is the one I would say is an expert in venom of old world vipers. He was with Benjamin Hempel studied the venom of Vipera moditis, and throughout Twitter again we started communicating, and we agreed to start the collaboration. This led to my first short-term scientific mission. I was welcomed in the lab of Professor Roderick Susmuth, where I spent 3.5 months in the Technical University of Berlin. And this experience, under the supervision of Mike, who is with a gigantic knowledge and really talented person to share that knowledge, I learned how to perform the bottom-up snake venomics protocol, so the full decomplexation of venom through HPLC, SDS page, triptych digest of the peptides, and then going back and assembling all those information into the 7 different venom pools that we analyzed. I got, as I said, the hands-on experience in the labs, learned how to analyze the data. And as a result of this collaboration, there is currently a manuscript that is under review. And about these results, I will talk a bit later. So for the continuation of my PhD, I was interested in studying the geographic variation of Vipera moditis. So with friends and colleagues, and with borrowing of the car from my parents, I did this tour around the Balkan Peninsula to collect the venom samples of moditis. And by the end of last year, I came to the lab and I was welcomed by Benjamin Florian Hempel at the Freie University in Berlin, in Germany, where I stayed for 2 months. And during that time, I did a characterization of all these venoms that were collected by using the MALDI top-down approach, as well as the venom on a chip fingerprinting. This is currently under analysis, and there is an idea to prepare 2 manuscripts based on these based on these studies and based on these results. And during this time, I had one more great opportunities to attend the Congress in Hamburg. So it was the Congress organ on the interdisciplinary summer school, actually, on snake bite envenoming. And here I had a 5 days opportunity to interact with people from different fields, from biology, chemistry, medicine, even anthropology. And there I discussed, well, I met Professor Wolfgang Wuster, and I discussed with him the, let's say, difficulties and opportunities in studying the venom variation. And then he suggested me to get in touch with a lab that I already knew from the publication, but I was not having any kind of contact with. So he put me in touch with Jeroen Kuhl and his working group. So during my, again, the STSM in Berlin, I took one day to go to the lab of Dr. Jeroen Kuhl, where I met also Julian Slagboom and Matthias Bittenbeider. And there I was introduced to the protocols that they were applying in their lab, and also with the new protocols that at the moment they were developing. So we agreed to form a collaboration and to analyze the venom of Ipramoditis using the venom variation venomics. And in general, the group is highly focused on developing the automatized or almost fully automatic protocols. So this would be an amazing way to assess the diversity of the venoms that I have. So hopefully, let's see if this web of collaborators will increase a bit more with some southern people from Southern Europe. So this was end of 2023. I'm still highly included in cost action. And then by the beginning of 24, Ronald came and say, hey, we're working group 5. One of the deliverables of this group is to prepare the syllabus for the master course, as well as to develop the Venomics virtual training program. I was interested in that, but I was not really sure how and if I will be able to in any way contribute. But then this scaled up. It included, so this program was led by Anna Klompen and Samantha Nixon as well. It was funded and promoted by Mande Holford, her funds from NIH Pioneer Award, as well as co-funded by Juven, therefore with many people who from the Juven organization, like Maria Vittoria Modica, who helped to actually have this joint work between people from the Gordon's Research Seminar and Mildred platform. And with all of this, there are some things that I'm now dropping like this. But so the Gordon Research Seminar is under the scope of the Gordon's Research Conference. The idea that is a huge conference on venom evolution, function, and biomedical application that was held a couple of weeks ago. And the idea was to present the 3 modules, transcriptomics, genomics, and proteomics module, to offer the data that was already published and to give the bioinformatics tools to participants to be able to learn how to process the data. Jeroen Kuhl took the lead on the proteomics module. In some collaboration, we agreed that I can help a bit in just do a small contribution to this proteomic module. So I was lucky enough that I had an access to learn how to prepare the, well, there was many things that were done throughout this workshop, and this workshop was given in the Gordon's Research Seminar. Sorry for now, a bit of a confusion. What is the next step is that actually, again, based and thanks to Juven, I was able to go to and be present there in person. I was awarded the ITC conference grant. So 3 weeks ago, I was in Portland in USA, first in the Gordon's Research Seminar, which is a 2 days event for the early career researchers, where I had an oral communication. This was followed by the 5 days conference, where I had a poster communication. And for me, this was the first ever Venom conference in person. And I was having high expectations, but this over-exceeded. It was absolutely amazing to see how people are open and how discussions are flowing and how everyone is approachable. So I really appreciated the opportunity to be there. Many new ideas appeared, and I think also collaboration opportunities were formed. And then I would say, I hope so, that also some new friendships were formed during these couple of days of intense living together and discussing the venom biology in general. So as we all know, the conferences are highly important, and I'm looking forward to meet you, to at least some of you, in a couple of weeks in Napoli, where there will be the second International Congress from Juven. There I will give a talk about the results of my first short-term scientific mission. And again, I hope that we will be, there will be a possibility that we all talk in person. And now actually I will go a bit more to the scientific part and present you the results of the first STSM, which is focused on the comparative venomics of insular and mainland Vipera moditis. And I will just give a second to. So this was done in, I would say, again, great setup, because there is a population in North Macedonia, in Prespa Lake, of Vipera moditis on island and in the nearby mainland populations. This means that these two populations are under similar biotic conditions. Climate and habitat are almost the same. However, these two populations differ in one really important aspect, and that is the prey availability, meaning that Golemgrad Island is totally depleted of small mammals, and vipers there are preying exclusively on ectotherms. This probably led to these vipers being also dwarfs, so they're somewhere 20% smaller than their mainland counterparts. In terms of the ontogenetic shift, so in the island, they are preying on centipedes and lizards. Throughout their whole life, they are having these two prey types as the dominant prey types. However, when they are babies, when they are juveniles, they are taking mostly centipedes, and as they grow, they include more of or try to prey more on the lizards. In the nearby mainland population, we have a control population where there is an ontogenetic shift, which includes mammals as they grow up. So here we could ask and hopefully answer the questions, are there differences in venom composition that are sex, age, and population related? And all with the idea to actually understand how do biotic conditions influence snake venom variation at the interspecific level. So to do this, we first did analysis of 58 individual venoms, separating it on the SDS page. And based on the gels obtained, we built up presence-absence metrics that was further used in different statistical analysis. For the more in-depth studies of these venoms, to do the identification of protein families and identification of the protein families itself, 7 venom pools were formed. So the ones that were presenting males and females, and all age groups in both populations. So what we got in terms of the venom composition, there are 10 toxin families. They are dominated by PLA2, SVMP, and SVSP. And what is a bit different than the other Viperina venom is that there is a high abundance of disintegrins, while C-type lectins are much less present here. This is something that I find interesting. I'm not sure. I would like to be able to explore it further, especially in the terms of the functionality of these disintegrins. But at this moment, we can just say that they are a bit different than the other Viperina venom currently known. As for the sex-related differences, well, in terms of the individual venoms, as you can see on this graph, the NMDS graph that is shown here, there are these venoms are highly similar. So there is almost total overlap in terms of the males and females venoms, which is also confirmed on the level of the proteome. So there are just minor differences. However, with this almost full overlap, we decided to combine the following age groups. In the following age groups, so the results that you will see later, there are males and females together in a venom pool. This is something that we expected. So because there are no differences between males and females diet, we were also not expecting to find any differences in venom composition, and we confirmed that in a way. Then for the age related, this is, as you can see along this NMDS plot, along the NMDS axis 1, there is a clear separation of juveniles and then 2 other age groups. So this shows that the juveniles are the most distinct group of venom phenotype. And this is confirmed at the proteome level, where we identified 2 venom phenotypes in juveniles. As you can see in the red, the SVMP is highly dominant, and Kunitz type, Kunitz is totally missing. As they grow older, Kunitz is starting to be expressed, and the levels of PLA2s and SVMPs are more balanced. So we confirm there are differences in venom composition between individuals on distinct body sizes. In mainland, there was a bit of problem. Okay. In mainland, there is an ontogenetic shift in venom composition, and this ontogenetic shift in venom composition is most likely linked to diet. However, in the island, we were not expecting this. We were expecting to find more of a pedomorphic profile of adult venoms, because since there are no small mammals, we were thinking that they will retain more or less the same composition as the juveniles. However, this is not the case. So what could be in the base of this? Maybe since we know that there is in one protein family, there is still high diversity of proteoforms of homologs, of post-translational modifications. Actually, maybe we are overlooking certain differences that using these techniques we did not see. So maybe these venoms can be explored a bit further. Then we can also see what are the actual biological activity of specific components. Maybe there are differences between the island and mainland populations by testing in vitro enzyme assays. And also something that would be nice, and I would like to do, if it is possible to do, to understand the functional consequences, because of these venoms are more potent towards scolopendras in islands, since centipedes towards in islands, since they are the frequent prey type, while maybe they are not at all active from the mainland population. So this is something that can be set up as a further exploration of the venom profiles. Since still these differences between island and mainland were something that I wanted to try to see if there are maybe some differences. We remove the juveniles, as they are the group that is taking the most of the variation when we analyze the whole system, the whole not the system, the whole sample size. And we just focused on the older age groups from two populations. However, you can see here still there is a full overlap, which means that there are no differences in venom composition between island and mainland vipers. This also leads to say that we can maybe say that the venom is well conserved in the same age group, even in the presence of distinct prey availability. And now this maybe, what is this potentially due to? Maybe there is a gene flow, dispersal of animals between these two populations, meaning that maybe some animals are coming from mainland and diluting the effect of specialization. Although there are currently no any genetic studies to prove or reject this, I think this is highly unlikely. So these vipers are small. They are like 60 centimeters of length, and I don't see them easily swimming the distance of 4 kilometers to reach the Golemgrad Island. But again, since there are no studies, nothing can be ruled out. In my opinion, that most likely the isolation time, or the time that these vipers spend on the island, was not sufficient for the specialization to occur. Also, there is one more thing that we don't really know. We know when more or less the island formed, some 5 million years ago. So it's one of the ancient lakes, not island, sorry. The lake itself 4 or 5 million years ago. It is a very old lake in Europe, but we don't know when the island formed. And also we don't know if the moment when the island formed and it was colonized, or the vipers appeared there, maybe there were also small mammals in that time. So maybe they were eating the small mammals for some time. They were having more of a standard diet, but then in some moment, for reasons of maybe prey, the lacking of resources, small mammals disappeared, and then in the evolutionary scale, these vipers are having restricted diet for a very short time. More questions that maybe will be answered in some moment. So I will leave you now with this more local study, and I will just want to take a zoom a bit and take us to the broader distribution of these species, where we can compare the results from our study, shown in green, and the ones from the Turkish populations. And here I would say there are striking differences. As you can see, there are no disintegrins in the proteomes. They are totally lacking in the proteomes of Turkish vipers, and there are high differences in other dominant components. So I think that all of this gives a good reason to study the venom composition of these species across distribution. And I hope that next time that you see me, I will be actually able to talk with you about the results from the distribution from the venom variation across the distribution of the species, and that we will see what is going on there. So with this, I would like to thank all of you for the attention, and I would like to thank Juven for all the opportunities that I was given to explore the venom of these species and to meet all these people that are actually Juven, because all of you are Juven. And I would say that I was really lucky that the cost action was aligned perfectly with my PhD, so I can learn a lot from everyone. And with this, I would finish my talk, and I'm open to any questions, which I hope I will be able to answer. Thank you.
Thank you very much, Margarita. Excellent talk. I will clap in the real world. That's always better than these fake yellow cartoon heads. Your journey from the beginning of your PhD during pandemic times, which must not have been easy, to now is just a shining example of, I guess, why Maria Vittoria wanted to do this action in the first place. I know she's in this in this lecture, and Gregor as well. You're just a shining example of how you can be a participant, a producer in your participation with the online training course, a consumer of the various possibilities, and an illustration, really beautiful illustration of how important it is to talk to colleagues, to do research these days. Because in the olden days, and that includes people like myself, some of us still like to sit in a room and do stuff on their own. But if you really want to do world-class research like yourself, you just illustrate how important it is to be mobile nationally, internationally. So I think there was a really nice illustration of the importance of this network. So that's really cool. So thanks for that.
Thank you.
Anybody with questions or comments, this is your chance.
Yes.
Yes.
So, Maggie, great talk, very interesting, and good to see that. I mean, we talked about your research a bit when we met in Hamburg almost 2 years ago. No, 1 year ago.
1 year ago, yes.
Anyway, a long time. So you said that you're interested in looking into the functional consequences. So if that is something you want to pursue, reach out, because that is what we do a lot, and especially all the enzymatic assays for Vipera we do very regularly. And Ignacio is about to show that just in a bit. So feel free to drop an email about that, because we do that anyway. I have one thing that I wanted to ask. I was interested in the fact that there is not so much venom variation between the populations. Of course, I believe there is certainly gene flow. It's not that far away. Vipers can and certainly do swim these distances, especially in this is not seawater, right?
No.
Yeah, so I believe there is gene flow. This could explain it. But one thing I was also wondering, have you considered that the prey, the centipedes, are just lacking a lot of the targets of viper venom? Because most viper venoms actually target the coagulation cascade, which is simply not present in centipedes. So if there's no target, there's also probably not so much evolution on these things, right?
Yes. Also, the thing is with the centipedes, there is probably not again enough time for that venom to specialize on centipedes, and that we can also see this because of the animals that were being that were preying, so the vipers preying on centipedes, which is also venomous and highly dangerous prey, and then the centipedes would eat out from the viper, which means that the venom probably did not subjugate fastly enough and in a specific way the centipedes. So could be that, yes, definitely there are components that would be present in specialized viper venoms that are for the invertebrates are lacking in this one.
Yeah, so I have so far not tested centipede venom or snake venom in centipedes, but we tested a lot of snake venoms in a range of insects. And what we see quite often is that viper venoms do not have a reasonable effect there, because the insect hemolymph at least is full with different protease inhibitors, and with venoms that are heavily loaded in proteases, of course, and you see not so much activity. So I think this is also something to consider at some point. So we would need to study the hemolymph of centipedes a bit. Maybe Ronald knows more about it than I do, because I'm clearly not a centipede person. But yeah, very interesting.
But even with the Vipera orsini, because they are specialized in the, those ones are maybe
this is one of the snakes we have not tested so far.
Okay. That would be nice. On that maybe I can help. But yeah, sorry.
No worries. We have a hand up, Lorenzo. Would you like to ask a question?
Lorenzo.
Hi Maggie again, like
Hi.
Tim said, really amazing talk, and yeah, I was already hooked on the Golem Grad project. Now I'm even more so. I have a lot of questions, so I'll get back to you later. But let's starting with the ontogenetic again, the ontogenetic venom shift, because I mean we call them ontogenetic shifts, but they are more about size than they are about age. So since you mentioned that the vipers on Golem Grad are like 20% smaller than those on the mainland, could it be that the shift is simply not triggered because they don't get to a size that would kick it into action?
The thing is, they are smaller, but what we are seeing that when the more or less the size when they are changing the venom composition in the mainland is the size that is also present in the island, in a sense. So around 30 centimeters, if I remember correctly, a bit maybe less, there is this shift in the venom profiles, and the vipers on the island reach 55 centimeters is the maximum. So let's say the average is 46. So it is included in the sizes are present in the island. So maybe it's not but I see your point that it maybe would not, that they don't actually reach the sizes where they shift. In this case, I think they do, that the ontogenetic shift is happening at a lower SVL body size, and that body size is present in both populations.
Well, that solves it. Thanks.
Thank you. That's a cool question. As any good talk does, it raises more questions than it answers questions.
Yes.
I wanted to say, in the interest of time, we should move on to our speaker. Our speaker has his hand up already, so if you have a question, go ahead, Ignacio.
I do, yeah. So nice talk, Maggie. Absolutely, I really like when there's always studies about venom variation. It's kind of my thing. I really enjoyed it. Following what Lorenzo said, it just came to my mind, this thing. Like, do you think that the reduction in size of the vipers on the island is adaptation, or rather like dictated by constraint due to a less, let's say,
Less energetic
diet? Yes, exactly.
Yes,
because in the end, if
definitely.
these animals are only eating scolopendra, centipedes, and have a venom that is not particularly effective at killing them, I would imagine that probably they don't have that much of an energetic input, is it?
Yes, they are definitely deprived of the energetically good, rich prey. And I think that if we would take some vipers from the island and feed them with a more standard diet, that they will grow there. I don't think it's a current constraint, and current in a sense, we don't know how long they are constrained, but it is, I would say, mostly energetically.
Could it also be due to population density? I mean, you told me there's like, in technical terms, a shitload of snakes on this little island.
Yes, it's probably one of the most dense populations of vipers in the world. It's like the estimation is that there are some 1,500, 600 snakes in an area. So the island is, I did not say that, but let's put it 700 meters by 450 meters. It's a tiny island with so many different. Also, they don't have predators. So maybe it can also be the constraint by the population densities.
Oh, yes. Let's not forget, I mean, it was an ancient colonization. This island formed millions of years ago. We don't know at this point exactly how long the snakes have been there, right? And body size decrease is an absolutely common phenomenon for both ectotherm and endotherm creatures on islands. So the factors that influence this are probably multivarious, but interesting. Anyway, Ignacio, I know that you're itching to speak more, and you will get the opportunity right now. Maggie, thanks very much for your talk.
Thank you so much.